Welcome back to our channel. Today, we're diving into one of the most groundbreaking discoveries in the history of biology, DNA. The double helix structure of DNA revolutionized our understanding of life itself. Join us as we explore the incredible journey of its discovery and the profound impact it has had on science and medicine. Our story begins in the mid-19th century with Gregor Mendel, an Augustinian monk whose experiments with pea plants laid the foundation for the field of genetics, and his discoveries continue to influence biology today. Let's take a closer look at his remarkable journey. Gregor Johann Mendel was born on July 20th, 1822, in Heinzendorf, Austria, now part of the Czech Republic. He joined the Augustinian monastery in Brno in 1843, where he found the perfect environment to pursue his passion for science. Between 1856 and 1863, Mendel conducted meticulous experiments with pea plants in the monastery garden. By crossbreeding different varieties, he observed how traits were passed from one generation to the next. Mendel focused on 7 characteristics, including plant height, seed shape, and flower color. Mendel's experiments led him to formulate the laws of inheritance. He discovered that traits are inherited in specific ratios and introduced the concepts of dominant and recessive alleles. His work demonstrated that inheritance follows predictable patterns, now known as Mendelian genetics. In 1865, Mendel presented his findings to the Natural History Society of Brno, but his work was largely ignored during his lifetime. It wasn't until the early 20th century, long after his death in 1884, that scientists rediscovered Mendel's research and recognized its significance. Today, Mendel's principles form the core of genetic science. His pioneering work has enabled countless advancements in biology, medicine, agriculture, and beyond. From understanding genetic disorders to developing hybrid crops, Mendel's legacy is vast and enduring. Gregor Mendel's dedication and curiosity paved the way for the field of genetics, transforming our understanding of heredity, but the true nature of the genetic material remained a mystery. Fast forward to the early 20th century, scientists like Thomas Hunt Morgan and Oswald Avery made significant strides in understanding that genes are made of DNA. 2 key figures in the field of genetics, Thomas Hunt Morgan and Oswald Avery. Their groundbreaking research on genes and DNA laid the foundation for our modern understanding of heredity and molecular biology. Let's dive into their remarkable contributions. We begin with Thomas Hunt Morgan, an American geneticist who revolutionized our understanding of genes in the early 1900s. Morgan's work with the fruit fly, Drosophila melanogaster, provided key insights into how traits are inherited. Morgan and his team discovered that genes are located on chromosomes and that they are responsible for hereditary traits. By observing mutations in fruit flies, they demonstrated how specific genes correlate with specific traits, such as eye color. This led to the concept of genetic linkage, where genes located close together on the same chromosome are often inherited together. In 1933, Thomas Hunt Morgan was awarded the Nobel Prize in Physiology or Medicine for his discoveries concerning the role of chromosomes in heredity. His work provided crucial evidence that genes are the fundamental units of heredity, situated on chromosomes. Next, we turn to Oswald Avery, a Canadian-American bacteriologist whose work in the 1940s was pivotal in identifying DNA as the genetic material. Avery's experiments with the bacterium Streptococcus pneumoniae aimed to understand the molecular basis of transformation, where a harmless strain of bacteria could be transformed into a pathogenic one. Avery and his colleagues, Colin MacLeod and Macklin McCarty, conducted meticulous experiments to determine what substance caused this transformation. They isolated various components of the bacteria, such as proteins, lipids, and nucleic acids, and found that only DNA could transform the harmless strain into a virulent one. In 1944, Avery published his groundbreaking findings, demonstrating that DNA is the molecule responsible for heredity. This discovery was initially met with skepticism, but it eventually laid the groundwork for the molecular biology revolution. The work of Thomas Hunt Morgan and Oswald Avery set the stage for future discoveries in genetics and molecular biology. Their research not only explained how traits are inherited, but also identified the molecular basis of heredity. These foundational insights paved the way for the discovery of the double helix structure of DNA by Watson and Crick, and the subsequent advancements in genetic engineering, genomics, and biotechnology. Thomas Hunt Morgan and Oswald Avery's pioneering work transformed our understanding of genetics and DNA, shaping the field of biology as we know it today. However, it wasn't until the 1950s that the true structure of DNA was uncovered. Enter James Watson and Francis Crick, two ambitious scientists at the University of Cambridge. Their quest to uncover the structure of DNA was supported by crucial data from Rosalind Franklin and Maurice Wilkins. Franklin's X-ray diffraction images of DNA provided the key insights needed to unlock the double helix structure, particularly the famous Photo 51, provided the essential clues needed to unlock the DNA structure. In the early 1950s, James Watson, an American biologist, and Francis Crick, a British physicist, were working at the Cavendish Laboratory at the University of Cambridge. Both were determined to uncover the structure of DNA, the molecule responsible for heredity. Using Franklin's data, Watson and Crick built a physical model of DNA. On February 28, 1953, they proposed the double helix structure, where two strands of DNA wind around each other, held together by pairs of nitrogenous bases, adenine with thymine and guanine with cytosine. This elegant model explained how genetic information is stored and replicated. The specific pairing of the bases suggested a mechanism for copying genetic material, which was a crucial insight for understanding heredity. In April 1953, Watson and Crick published their findings in the journal Nature. Their paper, A Structure for Deoxyribose Nucleic Acid, is considered one of the most significant scientific publications of the 20th century. The discovery of the DNA double helix revolutionized biology. It paved the way for the field of molecular biology, leading to groundbreaking advances like the Human Genome Project, genetic engineering, and personalized medicine. James Watson and Francis Crick's discovery of the DNA double helix transformed our understanding of life itself, laying the foundation for modern genetics. The discovery of DNA's structure revolutionized biology. It paved the way for numerous advances, from the Human Genome Project to modern genetic engineering and personalized medicine. Today, DNA research continues to unlock new mysteries about our biology and the diversity of life on Earth. But the story doesn't end there. The implications of understanding DNA extend beyond biology. It's transformed fields like forensics, anthropology, and even our understanding of human history. The ability to analyze genetic material has given us new tools to solve crimes, trace ancestry, and study ancient populations. From diagnosing genetic disorders to developing gene therapies, the impact of DNA research on medicine is profound. Scientists are now able to edit genes with precision, offering hope for cures to previously untreatable diseases. The discovery of DNA's double helix structure truly revolutionized biology, opening doors to endless possibilities and fundamentally changing our understanding of life. If you found this exploration of DNA insightful, please give this video a thumbs up, share it with your friends, and subscribe to our channel for more fascinating dives into the world of science. Until next time, stay curious and keep exploring.